Hey everyone, welcome back. This is the second lecture in chapter 13. And in this lecture, we're going to talk about the three basic bacteriophage types. These are based on their relationship with the host. So the lytic phages, these phages are virulent. They're virulent and they cause disease and they will kill the host cell. The temperature phages, these viruses tend to enter the host cell and then they incorporate themselves into the host genome. So they become part of the host genome, but if they get, if damage starts to occur to the cell, then they can eventually move out and become virulent so they can enter the lytic cycle if they need to. Filamentous viruses are also virulent. So they do cause disease, but they tend to not kill the host cell. So the general strategies for the phage, they have to be able to replicate, and they have to be able to then enter new host cells. So you can replicate new particles and then lyse open the cell and then enter new cells. That would be an infective stage. A latent stage would be where the virus is incorporated into the genome and it lies dormant, but it can be replicated and it moves along with all of the host cells that actually can benefit bacteria or give bacteria new characteristics. And so this is just looking at if you have a productive disease over here or if you have that latent state. If you have a disease, you're producing more viruses, and those viruses will eventually lyse out of the host and the cell will die. In this filamentous phage, you're going to also release viruses, but the cell doesn't die, it just replicates, causing disease, but the cells don't die. And then if you're in a latent stage over here, this is that temperature phage, it can enter into the host cell and then it incorporates its own genome into the host cell and then replicates along with the host genome. And so now the host has specific characteristics that differ from the normal viral or the normal genome because now they have viral genes as well. So let's look at that lytic cycle first, or the lytic phage. So lytic phage is going to start by attaching to a host. So viruses are specific to their host. You have to have surface proteins that will bind to the virus, which is why viruses are specific to different types. Like you have enteric viruses that attack the digestive system. You have respiratory viruses that attack respiratory epithelium. So they have those receptors. And so here we see attachment. The bacteriophage binds to the cell, and then it's going to release its genome, so its DNA or RNA, into the cell. At this point in a lytic cycle, the genetic information takes over the cell's machinery and starts producing lots and lots of small phage particles. that are going to eventually become new bacteriophages. So the assembly, so synthesis is producing all those particles, assembly is putting those particles together, and then release occurs when the cell actually lyses open. Temperature phages can direct a lytic infection or can incorporate their DNA. So this is a lysogenic infection. So you've heard about the lytic infection, so now let's talk about the lysogenic infection. A lysogenic infection starts the same way as a lytic infection. You still have that phage binding and the phage releasing its DNA inside, but now the DNA is inside and this is a lysogenic phage, so it's going to incorporate its DNA into the DNA of the host. And now the cell itself is now going to be called, or the new structure is called a prophage, because now you have phage DNA incorporated into the genome of the bacterial cell. That prophage is going to be replicated with the new cell, and so every single cell is going to have that prophage. Now, if something happens, that prophage can excise itself out of the DNA, and then it can undergo replication in the lytic cycle and assembly and death of the cell. So here we have an example of why a cell might die with a lytic or with a temperature phage. Here we have lambda phage. DNA is excised from the chromosome about once per 10,000 divisions. So if DNA is damaged in any way, this is going to cause the lambda phage to activate proteases. The protease destroy the repressors, which allow the prophage to be excised out. And so this is known as phage induction. And then the phage leaves without being damaged. But the phage leaves and survives while the cell itself dies. So lysogenic cells are pretty impressive cells. They are immune to infection by the same phage. So if another bacteriophage tries to infect it, it can't do anything. Lysogenic cells or lysogenic or prophage cells tend to carry different phenotypes than their normal cells. And a lot of the times this new phenotype is what causes the diseases that are associated with the bacteria. So Clostridium botulinum in and of itself doesn't cause any disease. But when lysogenic conversion occurs, then it produces a botulinum toxin only with that phage inside of it that causes botulinism. And all of these are other examples of bacteria that have a prophage inside their own cell that causes them to become pathogenic. The last phage then is your filamentous phage. So this is a single-stranded DNA phage. It looks like a fibrous, so kind of like a spaghetti fiber almost, that enters the cell. It causes an infection, but it doesn't kill the cell. Instead, you have the replication of the viral DNA, but then the viral DNA leaves, taking with it part of the cell's own membrane to surround it. And then it can enter another cell. I'm going to stop here and we'll get into generalized transduction in our next video. Bye.